All right, let's jump right into today's explainer. Today, we are shining a spotlight on a truly masterful microscopic pathogen, Chlamydia trachomatis. But we really shouldn't just think of this as your run-of-the-mill bacterial infection. Instead, I want you to look at this organism as a stealth invader. It's basically a biological infiltrator equipped with an incredibly sophisticated toolkit to hijack our cells, hide from our immune system, and replicate right under our noses. So whether you're a medical student prepping for boards, a healthcare pro, or just someone who geeks out over microbiology, you are absolutely going to appreciate the brilliant, if highly destructive, machinery of this little bug. Let's pull back the curtain on this microscopic, covert operative.
Okay, here's our roadmap for this explainer. First, we'll meet the stealth invader. Then we'll break down the chlamydial life cycle. After that, we'll look at antigens and factions, followed by clinical infiltration. Finally, we'll cover the diagnostic hunt and finish up with treatment and eradication.
Section 1. Meet the stealth invader, introduction and classification.
So for decades, scientists actually thought chlamydia were viruses. And honestly, you can see why. They share some very distinct viral traits. They are obligate intracellular organisms, which means they absolutely must get inside a host cell to survive and replicate. You literally can't grow them in cell-free artificial culture media, and they are so incredibly tiny they can slip right through standard bacterial filters. But make no mistake, they are definitively bacteria. They've got both DNA and RNA. They have a cell wall similar to gram-negative bacteria, though, fun fact, they actually lack a peptidoglycan layer. They multiply by binary fission just like other bacteria do, and most importantly for us, they are susceptible to antibacterial agents. That's our trump card right there.
Now, because of its really unique genetic quirks, the family Chlamydiaceae went through some recent taxonomic restructuring, splitting into two primary genera. First, we've got the genus Chlamydia, which holds the major human pathogenic species, Chlamydia trachomatis. That is our main focus today. The second genus is Chlamydophila. That one includes C. cetacea, which you'll find naturally in birds and poultry, causing atypical pneumonia, and C. pneumoniae, an exclusive human pathogen behind community-acquired atypical pneumonia. While this whole genus split stirred up a bit of drama in the taxonomic world, for our purposes today, we are zeroing in strictly on the genus Chlamydia and the widespread human disease it causes.
Section 2. The chlamydial life cycle. Chlamydia has this wild ability to exist in two totally distinct alternating forms. Think of them as the armor and the factory. First up is the elementary body, or EB. This is the extracellular infective form. It's tough, it survives outside the host cell, and its one job is to hunt down a specific receptor on a host cell, like a squamous epithelial cell, and lock on. But once it gets inside, it sheds that armor and transforms into the reticulate body, or RB. The RB is the intracellular replicative form. And here is the craziest detail. The RB can make its own nucleic acids and proteins, but it cannot produce its own ATP. It is literally an energy parasite. It flat out steals the host cell's ATP to fuel its own reproduction.
So let's walk through this incredibly elegant 48-hour cycle. It starts when the infectious elementary bodies attach to the host cell and get taken up into a vacuole. To avoid getting destroyed, they pull a stealth move and prevent the host's phagosome from fusing with a lysosome. Genius, right? Safe inside, the EB transforms into that metabolically active reticulate body and starts dividing like crazy, by binary fission. This massive replication creates a huge inclusion body inside the cell. Eventually, having totally drained the host's resources, these RBs transform back into EBs. Finally, the host cell just bursts, releasing a brand new army of infective elementary bodies to invade neighboring cells, and the whole vicious cycle starts again.
Section 3. Antigens and factions, biovars and serotypes.
To understand how this pathogen does its damage, we have to look at its structural weaponry, its antigens. At the genus level, all chlamydia share a specific lipopolysaccharide, or LPS. This is crucial because LPS acts by inducing pro-inflammatory cytokines, like TNF-alpha. This aggressive immune alarm is actually what leads to the severe tissue scarring and fibrosis we see in chronic cases. Next down the line, we have species-specific envelope proteins, and finally, we get to the serovar-specific antigens. These are the major outer membrane proteins, or MUMP, which are encoded by the OMP-A gene. This MUMP is exactly the marker scientists use to type and categorize the different strains of C. trachomatis.
Based on those MOP variations, C. trachomatis is historically divided into two main biovars, TRIC and LGV. And within those, we are looking at 18 distinct serovars affecting humans. Serobars A, B, B, and C are primarily linked to ocular diseases, specifically trachoma, which is actually a leading cause of infectious blindness globally. Then we have serovars D through K. These are the oculogenital strains responsible for genital tract infections, which we're going to dig into next, as well as inclusion conjunctivitis and infant pneumonia. Finally, the LGV biovar includes serovars L1, L2, and L3. These cause lymphogranuloma venereum, which is a deeply invasive ulcerative sexually transmitted infection.
Section 4. Clinical infiltration genital infections. In human hosts, those serovars D through K are just incredibly prevalent. For males, C. trachomatis is the #1 cause of non-gonococcal urethritis, or NGU, driving up to 50% of cases. Symptoms usually pop up about 7 to 10 days post-exposure with discharge and painful urination. It's also the main culprit behind epididymitis in men. But in females, the stealthy nature of this invader is especially dangerous. Mucopurulent cervicitis is the most common sign, but if it flies under the radar and goes untreated, the infection silently climbs higher. It can trigger endometritis, salpingitis, which is inflammation of the fallopian tubes, pelvic inflammatory disease, and ultimately cause devastating scarring that leads to ectopic pregnancies or even infertility.
And here's the thing, the damage isn't always localized to the reproductive tract. This brings us to a really fascinating, though severe, systemic complication, reactive arthritis, which historically was called Rader syndrome. This hits about 1 to 2% of NGU cases, usually a few weeks after the initial genital infection. It's known for a classic triad of symptoms: conjunctivitis, urethritis, and arthritis. It most frequently targets young men, specifically those carrying the HLA-B27 genetic haplotype. What's happening is a massive misfire of the host's own defenses. The local chlamydia infection triggers a hyperreactive immune response that turns around and attacks the patient's own joints, like the knees or ankles. It's a textbook example of the stealth invader weaponizing our own body against us.
Section 5. The diagnostic hunt. Lab diagnosis.
So how do we catch it? Let's walk through the diagnostic journey in the lab. It all starts right at the point of care with proper specimen collection. From there, depending on what a clinic has access to, they might use traditional microscopy and cell culture, rapidly pivot to modern molecular tests like NEET, or use serology to hunt for an immune footprint. Let's quickly break down these 4 pillars.
Okay, the absolute make or break moment is step 1, specimen collection. Think back to our definition. These are obligate intracellular bacteria. That means if you just lightly swab the fluid or discharge, you are going to completely miss the pathogen. The sample absolutely has to contain infected host cells, so firm scraping or vigorous swabbing of the urethral or endocervical site is mandatory. Alternatively, for urethritis or cervicitis, a first catch morning urine sample is awesome because it naturally flushes out and captures a ton of those cellular secretions. And if we suspect LGV, a bubo aspirate is the way to go.
Once we get the specimen to the lab, an older but classic method is microscopy. Standard gram staining doesn't really help us here since chlamydia stains super poorly. But direct immunofluorescence, or DIF, is a total game changer. By applying fluorescent monoclonal antibodies to the sample, those antibodies bind directly to the major outer membrane proteins. This allows us to literally see the pathogen glowing brightly as distinct inclusion bodies right there inside the host cell's cytoplasm.
Another traditional route is culture. Since it won't grow on artificial media, we have to grow it in living cells, like McCoy cells. For a long time, this was the absolute gold standard. You incubate the cells for 48 to 72 hours, then stain them with Lugol's iodine. Because C. trachomatis inclusions have this unique glycogen matrix, they soak up the iodine and turn a very distinct deep brown color. But while it's highly specific, cell culture is ridiculously labor-intensive, technically tricky, and honestly less sensitive than newer methods.
Which brings us to the absolute revolution in diagnostics: NAT, or nucleic acid amplification tests, like PCR. Today, NATs are the undisputed heavyweight champions of diagnosis. They've almost totally wiped out the need for cell culture because they are exponentially more sensitive and highly specific. They are much faster and can detect even just a microscopic handful of chlamydial DNA copies in a sample. Plus, they can easily tell the difference between species and serovars by targeting specific genes. If you want to catch a genetic stealth invader, you look for its genetic fingerprints, and NAT does this flawlessly.
Our last diagnostic tool is serology. Instead of looking for the bug itself, serology looks for the body's antibody response to it. This is mostly useful for systemic infections like LGV or infant pneumonia.
Using the microimmunofluorescence, or MIF test, we can mix a patient's serum with serovar-specific MOMP antigens. If the patient has antibodies against chlamydia, they bind together. We then add a fluorescent tag, which gives us a bright, glowing positive reaction compared to a dark, empty negative one.
So just to do a rapid-fire summary of our lab arsenal. Microscopy, especially diff, is great for direct visual confirmation. Culture gives us high specificity but is bogged down by low sensitivity and high labor costs. NEETs, like PCR, are the ultimate champions right now, fast, incredibly sensitive, and the current gold standard. Finally, serology serves a very specific niche role, mostly helping us track systemic disease by hunting for antibodies.
Section 6. Treatment and Eradication.
Fortunately, for all its stealth, C. trachomatis is highly vulnerable to the right antibiotics. For uncomplicated genital infections, the fix is pretty straightforward. A single 1-gram oral dose of azithromycin is the highly convenient drug of choice, or a 7-day run of doxycycline works wonderfully. Now, for complicated infections like PID or LGV, we have to hit it harder and longer, usually 2 to 3 weeks of doxycycline or erythromycin. But here is the absolute most critical public health rule. You must treat both sex partners simultaneously. Because this infection is so often asymptomatic, if you don't treat the partner, the stealth invader just bounces right back and the cycle starts all over again. Oh, and since co-infection with gonococcus is super common, ceftriaxone is also often added to the mix just to be safe.
We have covered a massive amount of ground today, but I want to leave you with a really provocative thought to chew on. Think back to that life cycle. Sometimes, when chlamydia is stressed out by antibiotics or a strong immune response, it actually refuses to rupture the cell. Instead, it literally hits the pause button. It arrests its development right at the reticulate body stage, entering a dormant state known as a persistent infection. If this stealth invader can just hunker down, go dormant, and wait out our best antibiotic strikes, how is that going to force us to rethink future treatments? Are we ever truly eradicating the pathogen, or are we just forcing it deeper into hiding? It's a massive, fascinating question driving the bleeding edge of infectious disease research right now. Thank you so much for joining me on this explainer. Keep questioning, keep learning, and I'll catch you in the next deep dive.